This video will introduce the topic of regulation of gene expression. How both prokaryotic and eukaryotic cells regulate the expression of their genes is a topic that has fascinated biologists for much of the past few decades. The question is fundamental to understanding how cells respond to environmental signals and to changes in their environment, and also fundamental to understanding the process of development, how complex organisms like us arise from a single fertilized cell. All of our bodily cells arose from the fertilized zygote, a single cell, by the process of mitosis, meaning that all of our cells have the exact same chromosomes and DNA content. However, they don't behave the same, they don't look the same. Our brain cells make neurotransmitters, our muscle cells make muscle proteins, our skin cells make hair. Can you imagine what a mess it would be if our brain cells grew hair? So the cells have to turn on and turn off genes appropriately at appropriate times. And this has been one of the most fascinating questions of the past few decades, ever since we discovered that DNA is the basis of heredity. How cells actually regulate the use of DNA and determine which DNA sequences are expressed, meaning transcribed and then translated to protein, and how cells are able to change or regulate the turning on and turning off the expression of specific genes is a key to understanding much of biology. To begin to understand this process, we'll take a close look at the lac operon as a model for transcriptional regulation. And it's a great model because it features both negative regulation, meaning turning genes off, and positive regulation, meaning turning genes on. Here is a graph that shows how E. coli cells can turn on the expression of the enzyme beta-galactosidase, which is encoded by one of the genes in the lac operon. And it's expressed or turned on in E. coli cells in response to a specific environmental stimulus, a chemical inducer, which under natural conditions is simply the sugar lactose. Lactose is a disaccharide composed of galactose and glucose covalently bonded together. So in medium lacking glucose, but with lactose present, we see rapid synthesis of beta-galactosidase upon addition of lactose. So lactose is added to the medium at time 0, and we see rapid and high-level synthesis of beta-galactosidase. Now this makes a lot of sense for E. coli cells because they would prefer, as long as glucose is around, they prefer to metabolize glucose. So as long as glucose is around, they don't need to make beta-galactosidase. They need to make beta-galactosidase only when glucose is absent and only when lactose, which is a substrate for beta-galactosidase, is around. If lactose is around, even if the cell is starving for glucose, there is no reason for the cell to make beta-galactosidase. So only when there is no glucose and lactose is present, then the cell has a reason to make beta-galactosidase and does make beta-galactosidase. So the questions are, why doesn't the cell make beta-galactosidase, or how is it that the cell does not make beta-galactosidase when either glucose is around or lactose is absent? And under these conditions, how does the cell actually turn on expression of beta-galactosidase. So to understand this, we need to again take a look at the lac operon of E. coli. So lac-Z encodes the beta-galactosidase. Lac-Y encodes a permease, which is specific for lactose. So permease is a membrane protein that performs facilitated diffusion of lactose across the cell membrane. And Lac-A is a transacetylase, or encodes a transacetylase, which modifies lactose when it gets into the cell. So all three proteins are all involved in lactose metabolism, and so it makes sense then for the cell to turn on all three genes at the same time when it needs to metabolize lactose. And conversely, to shut down all three genes at the same time if there's no lactose around or if there's lots of glucose around. So being an operon means that all three genes are transcribed as a single messenger RNA, and when ribosomes hop on and translate it, they will make the three different proteins from this single messenger RNA. Okay. So you can think of these as the target genes. These are the genes that the cells wants to regulate. What we then see is the Lac-I gene, which encodes a fourth protein called the repressor. The repressor is not subject to regulation. It has its own promoter, which is not shown here, but it's what we call a constitutive promoter. Constitutive means it's made all the time with no change or very little change in the level of protein that's made. So repressor is made all the time. And then we see three sequences in the DNA which are non-protein coding. So these sequences in the DNA called CAP and P and O are just specific sites in the DNA. They have a specific sequence of nucleotides, but they don't make any protein. They in fact serve as binding sites for proteins. So who are these proteins? Okay. Well, the first protein is RNA polymerase, the enzyme that transcribes RNA from DNA. So RNA polymerase, as you call, always initiates transcription at a promoter. However, it really doesn't like the lac promoter and ignores the lac promoter most of the time. And this is because the lac promoter is just not a very good promoter. The second protein is repressor protein, the protein encoded by the Lac-I gene. Remember, it's always made. And its binding site is the O sequence on the previous diagram that we call the operator. Its job is to bind to the Lac operator, and it functions kind of like a bouncer or a guard. It guards the promoter, and its job is to keep RNA polymerase away from the promoter. However, it has a sweet tooth. When lactose is around, it will grab lactose and leave the operator. So lactose is actually an allosteric regulator. So it binds to a site on the repressor protein and causes repressor protein to come off DNA. Okay. So you can see that in the presence of lactose, repressor protein will not do its job. Our third protein is CAP. CAP's stands variously for catabolite activator protein. Uh, you'll see some diagrams labeling CAP as the cyclic AMP activator protein. And then some other papers call CAP actually CRP, the catabolite repressor protein, which I don't want to get you confused. We'll call it the catabolite activator protein, okay? Because its function is to activate transcription. And it binds to the CAP site on the DNA. So the CAP protein binds to the CAP site on the DNA. Now, in order for CAP to bind to the CAP site, the CAP protein must itself first bind cyclic AMP. So cyclic AMP is another allosteric regulator. Of the CAP protein. So both the repressor protein and CAP have their own allosteric regulator. So the allosteric regulator for the CAP protein is cyclic AMP. And cyclic AMP you can think of as an alarm molecule that's made by the cell when glucose levels are low. So when glucose levels are low, the cell makes cyclic AMP. The cyclic AMP then binds to CAP, and the CAP protein with cyclic AMP bound binds to the CAP site on the DNA to recruit RNA polymerase. So here is a diagram that shows all of that happening. Let's say there is low glucose, so the cell is starving for glucose. It's making cyclic AMP. Cyclic AMP binds to CAP, and then CAP binds to the CAP binding site. What the job of CAP's job then is to recruit RNA polymerase. It tells RNA polymerase, hey, here's a promoter that you need to bind to and start working from. And so it brings RNA polymerase to the LAC promoter, and RNA polymerase will now then transcribe the LAC operon at very high levels. So this is how induction of the LAC operon works. However, it is negatively regulated by the LAC repressor. So negative regulation means that a gene is shut down, or transcription is shut down. Negative regulation, shutting down gene expression. So the repressor then works or shuts down gene expression whenever lactose is unavailable. So if there's no lactose, the repressor binds to the operator site, and you'll notice that the operator site is sort of downstream and maybe slightly overlapping the promoter. So the LAC repressor then acts as an obstacle to RNA polymerase binding. If it prevents RNA polymerase from binding, and even if RNA polymerase binds, LAC repressor acts as a roadblock and prevents RNA polymerase from proceeding to initiate transcription. So here we see LAC repressor performing its bouncer role, okay, guarding the promoter and preventing RNA polymerase from binding. So LAC repressor then is our negative regulator. Because it shuts down gene expression. And here is a structural diagram of LAC repressor. It's actually a homotetramer. It's one protein that forms a tetramer. And you can see sort of the head part of the molecule binding operator sequences in the DNA. Okay? There's the bottom part of the molecule is responsible for the tetramerization. And then right about here are the binding pockets for lactose. So when there's lactose around, lactose binds to these pockets and then causes repressor to lose the ability to bind to the operator. So this is how we get induction. First, we have to have lactose available. Then lac plus the repressor protein comes free. So it's soluble, doesn't bind DNA. So now the promoter here is unguarded, okay, as long as lactose is around. This is still not enough to get high-level expression. The unguarded promoter will be transcribed at very low levels by RNA polymerase because remember, RNA polymerase does not like the lac promoter. To get full induction or high-level expression, we have to have low glucose. The cell has to be starving for glucose. It makes cyclic AMP, and then cyclic AMP will bind to CAP. The cyclic AMP CAP complex binds to the CAP site, which is close to the promoter, and then it recruits RNA polymerase to the promoter to initiate transcription. So here you have it, positive regulation by the CAP protein. And here is a picture of CAP, and when it binds to DNA, it forms a homodimer. Okay? And here, right here, are the cyclic AMP molecules bound, and so the CAP plus cyclic AMP complex is able to bind DNA, and like many DNA binding proteins, when it binds DNA, it causes the DNA to bend, which appears to be also part of the way that many DNA binding proteins or transcriptional regulators work. Okay, so we have seen that E. coli cells can regulate expression of genes through proteins that bind to DNA, to specific regulatory sites in the DNA. So it's not just the lac operon, it's all other regulated genes in the E. coli and all bacterial and archaeal cells. All regulated genes work pretty much through specific proteins binding to specific sites in the DNA. Okay. So what about eukaryotic gene expression? Well, that same theme of regulatory sites in the DNA that bind specific regulatory proteins carries over to eukaryotic gene expression as well. So the whole theme then that is universal is that we get transcriptional regulation by specific DNA binding proteins. And proteins that bind DNA and regulate transcription are generally called transcription factors. Where eukaryotes differ from prokaryotes is that eukaryotic genomes encode many more activator and repressor proteins. The human genome contains about 2,000 DNA binding proteins which regulate gene expression. And eukaryotic genes have much more complex promoters. They don't have a if you thought the lac operon was complicated with 3 different binding sites, eukaryotic promoters have many more binding sites for proteins. In addition, eukaryotic genes are regulated by sequence motifs, okay, protein binding sites called enhancers, that can be many kilobases, thousands of bases away from the promoter, the start site of transcription. And they can be also even downstream of the start site of transcription, not just upstream. And finally, since eukaryotic DNA are packaged into chromatin, remember they're packaged with nucleosomes and into chromatin, and the chromatin can be in varying states of condensation, the regulation or transcriptional regulation is also partly achieved by regulating the state of the chromatin, or its level of condensation. For a gene to be expressed at all, the chromatin region containing the gene has to be fully decondensed and available for access by transcription factors and RNA polymerase. So here is a diagram, a couple of diagrams of eukaryotic genes, of how transcriptional regulation might work for eukaryotic genes. The first shown in here is the simple model. It's sort of the simplest case. Here we have the start site of transcription, and this is the core promoter, the TATA site is where RNA polymerase binds. Well, actually some basal transcription factors first bind, and then they bring RNA polymerase to the site. And then you can have a couple of upstream activating sequences, so this is where activators will bind. And then silencer sequences, which is where repressor type proteins or negative regulators will bind. And so that's sort of a simple situation. Here is a more typical situation. Again, you have the transcription start site. Okay. Again, you have the core promoter containing the TATA site. Okay. And then you can have multiple regulatory elements where different transcription factors bind, and silencer elements where repressors can bind, and then we see multiple enhancers which are many kilobases away, and these can be either upstream or even downstream. So it's the combination of numerous proteins binding to nearby sites and even distant sites in the DNA that all factor in to whether a particular gene is transcribed or not.